and were on unceded lands of their territory. Kristen, I've known since my PhD days, which is a long time ago. She really, you know, while the Shortman, Heath, Villadangos, people were buzzing away on the mouse DCs, Kristen and Derek were buzzing away on the human DCs, and none of the markers quite matched. We didn't quite know how they related to each other. And I'm sure the human people were like, these mouse subsets. And the mouse people were like, they just haven't looked hard enough for the mouse equivalents yet. But one day people found what the equivalents were. And in fact, Kristen was really pioneering that aspect. And she was one of the first ones to find the equivalents of human DC1s when she was in Derek Hart's lab at the Mater. Kristen now has her own group at the TRI in Brisbane and has really pioneered not only the description of the human DC subsets, but their development, and also now how we can think about using them in the context of cancer immunotherapy. And she's been doing a lot of work there on really complex models to try and bridge that gap between mouse preclinical models with actual patient translational research. So Kristen, thank you very much for coming down, and we really look forward to hearing your presentation.
Thanks, Charlene. All right, how do I move this thing forward? So I thought I'd give you a little bit of a background and history as well from the field of cancer vaccines, because they're really starting to be trendy again. And when I first started with Derek, they were decidedly untrendy for a while. And I guess what we're really trying to do is in terms of therapeutic vaccines is establish long-lasting anti-tumour immunity, much the way we do against infectious diseases. Not only that, but to also induce tumour regression and overcome resistance and recurrence in cancer patients. But also we need this to be safe. And I guess what the pinnacle, the underlying concept of cancer vaccines, which is somewhat different to the vaccines against infectious diseases, is that we're really focused on generating the CD8 T cell response, because at least we think, and there's a lot of hard evidence for that, is that the CD8 T cell response is really crucial for tumour killing. And of course, we wouldn't be here working with this if it wasn't that the fact that we know that dendritic cells really underpin these responses. And so, as Charla mentioned, I've been working in dendritic cells now for over 20 years, and really focused on human dendritic cells and how we can enhance them or manipulate them to improve vaccine responses against cancer. So we've got lots of challenges to developing therapeutic cancer vaccines over and above some of the standard issues that we have with developing infectious disease vaccines. One of the really key critical things that I don't really want to focus a lot of time on, but it's really worth considering when we think about how we can actually develop these things, is that every patient's cancer is different. That's becoming more and more apparent now, with particularly with single cell sequencing technologies. Not only that, within the individual tumour mass itself, the cancer cells themselves are heterogeneous, so finding an antigen to target even within a single patient's tumour mass is a problem. And then we compound that problem that cancers evolve within individual patients over time as well. So even within the same patient, different metastases are likely to be have different expression of antigens, and that can change over time. It can change over treatment parameters. And so it kind of looks a little bit like we're stubbing a needle in a haystack. And I think to a large degree, finding neoantigens and shared antigens is starting to address this problem in terms of what can we actually target, what's the tumour, what's the immune response in the tumour is actually going to see. Sorry, fly there somewhere. And so I don't really want to talk about that today, but I really want to talk about how we actually manipulate dendritic cells, how we get the vaccines to the dendritic cells, and how we can maximise that response regardless of what that type of antigen is. And so cancer vaccines have been used forever, really. They've been used for 40, 50 years. And when I was a young PhD student, I first started training with Peter Hersey in Newcastle, and he was really one of the pioneers of vaccine concept. He was infecting melanomas with patient melanomas with vaccinia virus, and administering them. And they were some of the first cancer vaccines that were administered in Australia. And I guess that's where that sort of kicked off my career. I remember a guy called Michael Lotes came to visit us in Newcastle at one stage, and he was talking about these dendritic cells and how exciting they were and what they could do and the potential of those in vaccines. And Peter said to me, he said, if I was you and at your stage of your career, I'd be going and looking at those DCs. And so here we are. And I think, there's a lot of different vaccine constituents that are being used over the years. So you can inject just the naked peptide protein, tumor lysates are being used, various types of nucleic acids and more commonly now RNA vaccines. Doesn't matter which delivery system you really need or which you decide to use, they all need to converge on a DC at some point. And so that kind of led to the concept really over 30, 40 years ago and the discovery that we could generate dendritic cells from monocytes in vitro, of taking dendritic cells out and using those as the vaccine. And then, of course, all these different types of delivery vectors, you could either use indirectly by injecting in vivo or directly by sticking on the DCs and then administering those back. And so they've been administered in thousands of cancer patients, hundreds of clinical trials over the last decade. And they were, when I was a young postdoc, really in their heyday, they had, were considered to be the next frontier and be really exciting. And really they sort of started to become a little bit more lackluster and fall out of popularity with the increasing success of checkpoint inhibitors and things. And I guess now is really nice to be starting to come that full circle with the realisation that the checkpoint inhibitors and other different sorts of things actually aren't working they work really well when they do, but there's a huge amount of cancer patients that they don't work in, and we've still got to do other things to those patients. So I guess the best way to induce immune responses is to vaccinate. So they're really beginning to enjoy a bit of a renaissance again now. So what's out there? So I just thought I'd run through a couple of snippets of evidence that they actually work, and these are the few cancer vaccines or indirect or directly called cancer vaccines that are actually FDA approved. And the first one of these was the BCG vaccine for bladder cancer that's been around now for nearly 50 years. The reason it's called a vaccine really is because it uses BCG, which is obviously used to as a tuberculosis vaccine. But the BCG really specifically bind, or not specifically, but has increased binding to bladder cancer cells. And that induces not only lysis, but also provides antigens expressed on those bladder cancer cells that the immune response can see. And it's presumed that the DCs then take up those antigens and kickstart the immune response. So that's why it's called a vaccine. And has been enormously successful and FDA approved and continues to be used to this day. Another one, which is a kind of indirect vaccine that is also FDA approved, is an oncolytic virus called TVAC. This is FDA approved for malignant melanoma. The response rate isn't that great. It's, I guess, what we'd call an in situ or an indirect vaccine, in that the oncolytic virus then infects or more specifically targets the tumour cells and causes tumour lysis. And again, that gives the dendritic cells something to see and present and be activated by, and resulting in immune death. So that's another slightly different type of cancer vaccine that's also FDA approved. And just to shout out now, because I'm sure everybody's aware of the field of RNA vaccines and how they're taking off. These are already really well developed and had already been tested in phase 1 trials when COVID came around, and I think we've got a couple of decades of people messing around with RNA to try and use that as vaccines for cancer, to thank for how rapidly those vaccines were deployed in the case of COVID. And so now that's really accelerated this field and enabled these vaccines to really move ahead for cancer. So there's heaps of clinical trials going on. All the big players have these trials, and they're looking really, really good. And so one of these is the Merck Moderna vaccine, which is a personalised vaccine. of neoantigens, and that's just received fast-track FDA approval and is now going into phase 3. So, and you'll note there's 1,000 patients that they're intending to recruit over the next 5 years. So again, very, very exciting. And again, another concept that, that these things still need to get to a dendritic cell for maximal efficacy. And this is the first FDA-approved cell therapy for cancer. So this was approved prior to all the CAR T cells, and this is also considered, I guess, a dendritic cell or a cellular therapy-based vaccine. And this is called Provenge or Sipuleucel T from Dendrion. The idea is that the leukocytes are harvested from the patients and enriched and made into these dendritic cell-like things that are then reinfused into the patients. At the time that this was FDA approved, nearly or over a decade ago, they were $100,000 per patient. Dendrion went broke, and it's still being offered as a therapy, but it's really the logistics and the practicalities of growing these things, as well as the very modest, I would say, improvement in survival have sort of left this hanging on the shelf for a little while as well. But there is also this other one that is now in phase 3 called DC Vaxel, and that's for glioblastoma, and that's using tumor lysate, patient-specific tumor lysate loaded onto dendritic cells. And again, that's looking very encouraging in larger phase 3 studies. So there is some pretty good evidence that doesn't, regardless of the type of approach of vaccine delivery, that both dendritic cell-based vaccines and indirectly dendritic cell targeting vaccines in vivo can have some efficacy in cancer. And clearly we've still got a long way to go. So when we started entering this field, all of these trials and everything were done with monocyte-derived dendritic cells. And Derek, who I was early postdoc with, was really one of the pioneers that started looking at these monocyte-derived dendritic cells and looking at the ones that we found in peripheral blood in patients and saying, well, hang on, these are different. There are some similarities that are different. And so he was really a very firm advocate of using the natural circulating dendritic cells rather than the monocyte-derived dendritic cells. And so I spent probably, 10, 15 years of my life sort of is really sitting in this slide, and Melinda Hardy, who's in the audience, put some blood, sweat and tears into this as well. So at the time we were defining dendritic cells. We could see naturally circulating dendritic cells in blood. We use this marker called BDCA1 to define them, which is also known as CD1C. And so Miltenyi had devised clinical grade isolation systems to be able to isolate these things. And so we spent a lot of time looking at these, directly comparing them to monocyte-derived dendritic cells, showing that they were at least as effective, and if not in several cases more effective than monocyte-derived dendritic cells in terms of priming tumour antigen-specific responses. We looked at them in cancer patients and really looked at their how functional they were and their phenotype. And yes, there were some functional deficits, but the bottom line was that we could get these things out. We could get them out in sufficient numbers and sufficient function to actually justify taking this to clinical trial, and that's exactly what we did. So this was a phase 1 clinical trial where we isolated these blood DC1 dendritic cells directly from patients. The advantage of our trial, that was running about the same time as the phase 3 Dendrion trial was running, we could get 82% purified dendritic cells, whereas the Dendrion trial, they were running at best 10 to 13% dendritic cells in their vaccine prep. It was highly feasible. It was a small phase 1 safety study, but we could manufacture that vaccine from all 12 patients that were entered into the trial. And really importantly was a very quick turnaround time. So from taking the leukapheresis product, we could re-administer our vaccine within 24 hours, whereas the Dendrion one was shipped off to a site for manufactured for 3 to 5 days and then shipped back. So we had some really key advantages and advances in terms of targeting or harnessing naturally circulating dendritic cells over monocyte-derived dendritic cells. The vaccine was safe, well tolerated, so it really met its phase 1 endpoints. We found some evidence of immune responses later on, but in the end we didn't proceed to phase 2, mostly for funding reasons and other institutional reasons that we never got to progress with that. But I guess I can tell you as a postdoc as well that really almost was a career killer. You don't get Nature papers from fiddling around with patients' blood and working out how to isolate and study DCs. And so it sort of sent us back to look at these things in more detail. One of the other subsets that were sitting in there, we thought they were 0.03% of peripheral blood. We thought because they were so low that they weren't important. We didn't even really consider that they would actually be a real and distinct subset. But while that trial was going on, we started going back and having a closer look at those in more detail. And they were cells that we defined by the marker of CD141 or DC1. And we started, we would need a leukapheresis product, we would need a 16-hour isolation procedure to get enough of these cells to just have a look at them. So this was a really hard slog to do some of these studies back in the day. But one of the really key findings that we found from this, we'd set up these cross-presentation assays, and this was viral antigen. I won't go through the details, it's old data and it's published. But what we found is that monocyte-derived DCs that everyone uses for trial were really not very effective at cross-presenting dead cell antigen, which is the key response we need for tumour immune responses. And our CD1C DCs that we, see that? Our CD1C DCs that we had shown to have promising clinical trial did a little bit, but they didn't do anywhere near as what the CD141, this really rare silly subset, was doing. And it kind of told us two things, was that, you know, this rare subset, despite how rare it is, that it must be pretty important, but it was also really the first indication and really the first time we put forward this hypothesis that maybe they're the ones that we need to be looking at and targeting if we wanted to improve immune responses and vaccine approaches for cancer. And so that's what we started looking at. And of course, we weren't the only ones to be looking at in this space, and with the discovery of more molecules and being able to microarray technology and things came into place, we were then able to start aligning our subset with the mouse, what we now know as the mouse DC1, or collectively known as mouse human and mouse DC1s. And of course, in the mouse, they were defined as CD8 and CD103. And the really key things that moved the field forward were finding this CLEC9A, which was discovered here at WeHi, which is a dead cell receptor that's specifically expressed on both human and mouse DC1s. This other molecule, CADM1, which is, we still don't really know a lot about it. It's an adhesion molecule. Its ligand is co-expressed on activated CD8 T cells and NK cells, and thought to help facilitate the interaction between those two cell types. And their other key defining molecule on their surface was this chemokine receptor XCR1. And again, its ligands XCR1 and 2 are also expressed on activated CD8 T cells and NK cells. And so collectively now we know that these cells are really specialised for interaction with cytotoxic lymphocytes, both CD8 T cells and NK cells. They're also enriched in TLR3, and we know that when we activate them with poly-IC and other TLR3 ligands, they make huge amounts of interferon beta and interferon lambda. And a couple of things really to really emphasise that the need to study human cells as well, even though there is this general homology between mouse and human DCs, there are still some key differences as well, and some of these could be really, really important when we consider how we're going to harness these for vaccination. And a couple of those that we and others have identified over the years, sorry, go back, here. So We were using CD141 in humans to find them. That's not expressed in the mouse. Human DC1s also express TLR8, and so that gives you a whole a whole repertoire then of TLR8, like ligands and things that we can potentially use to activate those cells, and that can't easily be studied or modelled in the mouse. And the mouse equivalents express both TLR4 and TLR9, and actually a lot of the adjuvants and things that have been used or being looked at in mouse models are really that focus on targeting these TLRs are really going to be difficult or need special consideration if you're going to try and translate them, because they're just not there on the human cells. And a couple of other key things, they do produce IL-12. They can produce a lot of it in the right circumstances, but so can the DC1s or the other human subset, and that's a really key difference between mouse and humans. And also CLEC9A, that it's really exclusively expressed on DC1s in human, but it's also expressed on PDCs in the mouse, and so we need to be mindful of that when we're considering targeting technologies with CLEC9A. And this is a really nice figure that Florent Junot published just this year, that really summarises where we are right now in terms of human DC subsets. And a lot of this is really now starting to become apparent when we're starting to look at single cell sequencing of tumours across lots of different human tumour types and align those with the mouse. And really consistent with their role in CTL induction, the DC1s have really got a key role in tumour immune responses, and that's been shown in multiple different mouse models and multiple different tumour settings now. The DC2s are probably important as well. These are what we were calling the CD1C DCs, or the BDCA1 DCs in humans. We don't really fully understand this yet, but they are proposed to be driving CD4 T cell responses, and even in some human systems, there's very clear evidence that they can drive CD8 T cell responses as well. Both of these subsets now have also been shown to induce this, this DC state in human tumours that we call MREG DCs now. And that stands for mature regulatory DCs, and that's because in the tumours they've got both maturation molecules as well as regulatory molecules. We really don't understand much about these, how they develop, why they develop, or importantly what their function is, and whether that this is actually a tumour-inducing anti-tumour state or a regulatory state. And so this is a really right area of investigation that we're looking at. And in contrast, we've got these things that is really the camp's out at the moment, whether they arise from a classical dendritic cell progenitor or whether they're a monocyte derived. I think the latest is that they're probably monocyte derived. Who knows? They're very, very similar to monocyte derived DCs. And then, of course, we've got the plasmacytoid DCs. And so, ironically, to date, all of these types that are sort of going the wrong way towards immunosuppression are the types that we've been used for cancer vaccines. So it's kind of little wonder that they've had such lacklustre performance in the clinic so far. So there's been tons and tons of evidence. I'm not going to run over this, but not only are the DC1s important for inducing immune responses, they're also really important for making some of these other immunotherapies work. With the caveat that a lot of these studies have been done in the beta-3 mice that lack DC1s, but they also have some other issues, particularly with their CD8 T cell responses. But the bottom line is if you don't have these DC1s, you don't get a CD8 T cell response. And then any of those CD8 T cell responses treatments that are mediated by CD8 T cell responses, such as checkpoint inhibitors, your adoptive T cell therapies, even radiotherapy and oncolytic viruses that rely on that CD8 T cell response, don't work. And so the idea is then if you can target the DC1s, then you're going to get better responses to not only as a vaccine in its own right, but also enhanced responses to other immunotherapies as well. What we know about them so far is that mechanistically, and pretty much all of this has been done in the mouse, how they induce tumour immune responses. They rise in bone marrow from FLT3 ligand progenitors. and are attracted into the tumour site. They respond to damage, whether that's conventional therapies or other forms of cell death and interferon. And the classical paradigm is that then they become activated and they run off to the lymph nodes and talk to the CD8 T cells, which then come back. But we also know that they are playing a really important role at the tumour site as well. So I think the field is still really undecided which is more important or both at play. But there's certainly interactions, particularly the secretion of chemokines and things that then can attract those CD8 T cells back to the tumour site and can probably prime and reactivate the CD8 T cells at the tumour site as well. We know a little bit about how they're getting in, and there is some evidence that NK cells within the tumour site can attract these in either by expressing FLT3 ligand and various chemokines to bring them in. But we also know that the tumour's got a whole load of suppressive functions, both intrinsic and other, particularly myeloid suppressive cells that can stop this whole process. So better understanding of this, then we can really start to work out clever ways of really harnessing these cells to reinvigorate immune responses. In humans, there's a whole pile of evidence now to suggest that their signatures, not only in melanoma but in lots of other cancer types, are associated with good prognosis. So lots of DC1s at the tumour site, you're much more likely to have a favourable prognosis and survival, and you are also more likely to respond to immunotherapy such as anti-PD-1. And also there's lots of correlates of these, so they're correlated with enhanced CD8 T cell signatures and NK signatures. So a lot of good evidence in humans that lots of DCs at your tumour site is very favourable. And this is some of our work that we started doing, looking really for biomarkers and for understanding in cancer patients just really what their DCs are like. This is a study we did in melanoma a couple of years ago, where we developed a whole blood assay to look at, to really specifically quantitate DC numbers and also get some functional data from those in cancer patients. And what we could show that with stage in melanoma, even systemically, is that the DC1s, which are the CD141s down here, are significantly increased, whereas with increased tumour progression or stage, lots of the other cell types, including the other myeloid monocytes subtypes were increased. And the DCTs and the PDCs were really unchanged. So not only have we got defects at the tumour site, but we've actually got really systemic defects in their hematopoiesis and development as well. And we're not the only ones to show this. This has been shown in a few other cancer types as well. And also notably, if you, in this whole blood assay, if you activate them with different TLR ligands, not only are there's fewer of them, but they're less able to respond. And that's just shown here in terms of co-stimulatory molecules such as CD83 and MHC class 2, which is HLA-DR in humans. In that same study, we also tracked them over time in patients responding to anti-PD-1 immunotherapy, and what we found is that in the patients that didn't respond, the numbers are low anyway, and they remain low. The dotted line here is the healthy donor median. But in the patients that don't respond, they just continue to decline. Whereas in the patients that respond, we did see a little bit of a decline initially, but they sort of bounce back and stabilise. So again, further evidence that if you haven't got these DCs there, even at a systemic level, you're not likely to respond to immunotherapy. So there's a lot of ways now, and a lot of potential ways in which we can target them, both either in vivo or by adoptively transferring them in. And a lot of these studies have all been really well examined and show promise in a lot of mouse models. So we've got ways of expanding them. We've got FLT3 ligand, for example, that's been tested in hundreds of cancer patients. It's safe. There's no doubt it beats DC numbers, but we've got some problems with that too. As a monotherapy, it's been shown to be not very effective. There's that risk that if you increase lots of these DCs, they're also inherently tolerogenic, not just immature, but very tolerogenic. Lots of those without having to activate them is very problematic. So then you need combinations with activators such as poly-IC. The FLT3 ligand, there were two sources. So Celldex, that was really making a lot of this, has taken FLT3 ligand off its development program. So I think it's available for some ongoing clinical trials, but it's no longer going to be available. So that's going to be a bit of a problem. And a recent company, I can't remember who it was now, Gilead, made a more stable version, because the Celldex version you have to inject every day, which is also a problem to be used clinically. They made a more stabilised version, and their phase 1 has just been terminated for safety reasons. So that might be a bit of an issue for looking at trying to expand the numbers in vivo. So we've got to start thinking of other clever ways that we can do that. Likewise with activation, poly-IC or the stabilised analogue poly-IC LC is safe if you inject it locally. So if you inject it in the skin, for example, but you can't use that systemically. So that gives you limitations in what you can use that with. So ultimately, I think you're going to have to use some kind of combinations. The more of these combinations, the more problematic that becomes. We've also been looked at, and I'll show you some data from this in terms of improving targeting by targeting those the DC1s in the lymph nodes and delivering antigens specifically to them. I think a lot of similar things there in that these don't directly activate the DCs, so we need to look at more improved versions of these that can both activate them and potentially expand them, because if they're not there or they're defective and dysfunctional in cancer patients, we're going to have a hard job getting these things to work. And the other way, of course, that's of some interest to you guys here as well is this concept now that if we can grow them, that we might be able to adoptively transfer them. So adoptive transfer is not really going to work, I think, from just collecting, or we know it's not going to work from a patient leukapheresis product. People have tried. We spent years trying to do this. There's just not enough of them there. And as I showed you, they're not very functional either. So that's not going to be a good approach. But what we can potentially do is look at strategies that we can grow them from either a third party or from the CD34 progenitors in vitro. And I guess then we've still got all these same sorts of issues that you have if you're targeting them in vivo. So what do you activate them with? What kinds of cocktails? How are you going to grow them to GMP grade? Where are you going to put them in? Is the personalised vaccine actually going to be a go or not, or are we going to have to go to an off-the-shelf? And time to access, I guess, is an important consideration here, because sometimes cancer patients are not going to be well enough to take their own blood, or they're not going to be well enough to wait their, 2 weeks to 3 months to whatever it is it's going to take to make their own. So we've been really starting to pursue and strongly advocate for an off-the-shelf type therapy that we could potentially use, for example, from cord blood CD34 progenitors. And a couple of my students last year, just a couple of years ago, just published a really nice review that sort of talks about all those issues. So over the last decade, we've also started to look at ways that we can study these cells better in the lab, and we've developed a number of different ways, mostly focused on cord blood CD34s. We can now develop DC1s in large numbers and high purity, either from in vitro culture conditions or by putting these into immunodeficient mice in what we call humanized mice. And I'll talk a little bit more detail about those later. We can also start to genetically modify those expanded CD34s. And this has been a great tool. We do this mostly with lentivirus. We can get really good transduction both in vitro and in vivo, and that's just shown here in terms of the percentage of DCs generated after a lenti transduction in our lab, or the DCs that go on into humanized mice. And so this has really started to transform our ability to look at these cells, to be able to optimise vaccines and immunotherapies that target them, and just to understand their function in a bit better way. And that's just a selection of a bunch of papers from us and collaborative works that we've done with others that have harnessed these sorts of technologies to look at human DC1s. So the humanised mouse thing, I just wanted to digress and talk a little bit about that and why we got into that. I'm probably going to run out of time, but and I'm probably preaching to the converted anyway, in that, you know, we've got this huge discrepancy between mouse and human immune systems that don't always translate, and it's particularly problematic in the field of cancer, where we've got less than 8% of cancer drugs actually get to approval. Most of those fail because of lack of efficacy or toxicity, and so we really need better ways to bridge that gap. Particularly for DC1s, we initially started looking for a tool that we could look at larger numbers of them. And so humanised mice are really starting to become powerful models in bridging this translational gap, and we are just about to set up a first designated facility for generating humanised mice that will be available for the Australian research community at TRI. So it's been a bit slow. It's taking us a couple of years to get this off the ground, but we're hoping to be able to launch that early next year. So we've been using these for a bunch of different applications, and the idea is that you take CD34 progenitors, you stick them into some form of immunodeficient mouse, and that mouse then goes on to develop various components of a human immune system. And we've got a lot of broad-ranging applications, and we've been working on lots of these different applications with our different collaborators. Just to give you an example of their potential is this really infamous and very sad case of this drug called TGN-1412, which is a superagonist antibody that was safe and was done, in complete due diligence in all the in vitro models and non-human primates, and was cleared to go to clinical trial. And that had catastrophic and lifelong consequences in 6 healthy volunteers back in 2006. And that was, in essence, a cytokine storm that could not have been predicted from the existing models. And fast forward a few years after that, humanised mouse models have been used, could have predicted that, and are being predicted for those sorts of purposes, and are now being advocated for use by the FDA for those. So we think they're really powerful and important models. There's a number of ways you can generate these. I'm happy to talk about this a little bit later, if you like, to anybody that's interested. Our main approach is this approach where we take CD34 cord blood-derived progenitor cells and differentiate those. Other different approaches are to infuse PBMCs. There are limitations with infusing PBMCs. They're a great model for GVHD, and they're quite short-lived, and they're also mostly T cells that develop. So they're really good for some applications, but not necessarily for tumour applications. And the other one that's pretty much inaccessible to the Australian research community is what we call the BLT mouse, and that's where we have a human bone marrow, human cord blood HSCs from fetal liver, thymus, and kidney that are used, so the human cells undergo thymic selection on human thymus. So that's really good for human HLA-restricted T cells, but again, is really not accessible to us. There are a lot of limitations, and you really have to pick your model depending on the cell type. So some models are better for some types of human cells, and some models are better for others. And a lot of this is due to the different discrepancies in cross-reactivity of cytokines, etc. The models that we tend to use the most is the NSG-based strain that's transgenic for human HLA-A2. And that gives you the ability to induce human antigen-specific A2 restricted T cell responses. And we also use the NSG SGM3s, so they're transgenic for human IL-3 GM-CSF and SCF. And they're really good for increasing myeloid cell development, and then by default you get increased NKs and T cells as well. But there's lots of other strains, and this is a really rapidly evolving field with new strains emerging all the time. So this is our workhorse model. We take human cord blood CD34s, we engraft them into neonatal mice. We do this because those mice then have a thymus, and if they've got human HLA-A2, and such as the NSG-A2s, then you get development of human HLA-A2 restricted T cells in the human thymus. And from 10 weeks onwards, you start to get development of human immune cells, CD45 in most of or in all of the organs. What was really attractive to us at the time is that we get all of the DC subsets developing, and in particular the DC1s. And if we treat these mice with FLT3 ligand, we were getting as many DC1s out of a single mouse as what we were out of a whole leukapheresis product from a human. So that was really one of the main motivations for using these models in the first place. We've done a lot of work on characterising these, and by every different means that we can look at, both their phenotype, their transcriptome and their function, they really resemble those that we can see in human blood, particularly in terms of their capability to produce interferon lambda and also for cross-presentation. And this is just to show you some of our early work that they do align, at least by that kind of analysis. These are the humanized mouse DC1s here compared to human blood, so they do align pretty well. More recently, Zara Elahi, who is co-supervised by Kristen Wells just across the road, and I developed this lovely tool or atlas on the STEMformatics platform, where she's curated a whole bunch of human DC data, and she's been able to then you can project your data onto this atlas to see expression of various molecules. And for example, you can see the DC1s here expressing CLAC9A as we would expect. But you can use this as a really unbiased way of projecting your DC data onto here to see where they fit. And so we've looked at that with both of our in vitro and our in vivo DC1s. And one of the really key points to notice, which is really nice about this technology, is that the in vitro DC1s and in vitro DCs in general sit very far apart from those that are from human tissue DCs, or those that are directly isolated in vivo. And what Zara's been able to do is be able to look at the differences in these two, so that we can start to develop new in vitro culture techniques and new systems to bring our culture models more closely to what the in vivo DCs look like. And in our latest ones, which are on the DR1 feeders, which is a model based on some protocols by Strickland Ballen, both those in vitro DCs and our humanised mouse DCs they're not quite there yet, but they're not up here either. So they're kind of a step forward, and really trying to work out what those key differences is will really help us move those forward. We've done a lot of work looking at what optimal activators we should use for DC1s. Poly-IC we know is good and has been the gold standard and used in the clinic. We've got a lot of data that really aligns really nicely with a lot of the mouse work that's done, that shows that poly-IC is not enough. We're probably going to need to activate with anti-CD40. Some of this work was recently published, some of it is unpublished. But the key message is that all those cytokines and other molecules that you need for effective T cell priming, such as IL-12 and IL-27, TNF and IL-6, you're really only getting decent amounts produced by DC1s in synergy with either poly-IC with CD40. You can substitute poly-IC with type 1 interferon and type 2 interferon. Interestingly, they really make large amounts of interferon beta, but not so much interferon alpha, which is what we expect. They make large amounts of interferon lambda as well, but interferon lambda never synergizes with this. And so, really trying to pull apart these and work out what the best types of adjuvants to use going forward, or to target with these is an ongoing area that we're particularly interested in. So the other thing that we're starting to do now is get some more complex models where we build in a human immune system and then engraft in a human tumour. It doesn't matter whether these are mismatched or not. We're not the only ones to show this, but we see really good engraftment and tumour growth, either in the presence or the absence of that immune system. And so that kind of tells us that, similar to most cancer patients, that human immune system in those mice is either ignoring or being tolerized against against that tumour. And so that really gives us a good model then that we can start to look at immune interventions. And the tumours in these mice do get infiltrated with human immune cells. We get lots of T cells, but we also get lots and lots of, we don't know what they are, they're monocytic myeloid type 11B positive, 14 positive cells. We do see a little smidge of DC1s, but they're really hard to see in those models. So to sort of start to get some bones around whether we can actually target DC1s in humans and whether a lot of the mouse data stacks up, we've started treating these mice with different with different sort of DC1 direct or indirect type treatments. And so in this example, we've made a humanized mouse with a melanoma and a human immune system, and we've given it various immunotherapies. So one FLT3 ligand and poly-IC, which increases DC1 numbers and also activates them. Those DC1s do go into the tumour site. The other DCs do as well, but we get a significant increase in the DC1s in the tumour site. If you treat with FLT3 and poly-IC alone, we really don't have any significant effect on tumour growth. If you treat with anti-PD-1, which is pembro, again, you get a similar response. And we only gave a very short 2 doses of this, what we call the tritherapy, where we did the combination. But it was only after the combination there that we could see some evidence of reduction of tumour growth. So that was really the first evidence that doing these sorts of combination therapies, and particularly DC1 associated therapies, might be transferable to the human setting. And so we went one step further then and tried the same approach, but took the DC1s from a secondary mouse and then adoptively transferred those into that model and this was an intratumoral injection to ask whether DC1s could reduce tumour growth as well. And this was all done in combination with anti-PD-1. And what you can see from here is that the DC1s were able to, and this is just after 2 short doses, were able to have some effect on reducing tumour growth, whereas adoptive transfer of DCTs have no effect. And it's really hard to look into mechanistic details in some of these human studies, but from what we could tell is the DC1 treated mice were also had increased infiltration of CD8 T cells into the tumour site as well, so indicating that it was a CD8 T cell response. In that model, it's almost certainly allo, but that's where it is. How am I going for time? Wait. 8 minutes. 8 minutes? We started a bit late, didn't we?
Maybe 3 more minutes.
3 more minutes? Okay. We have looked at, and I'm sure you're all aware of the work that we have been doing in conjunction with Mirela Hood at Monash to translate her translate CLEC9A targeting antibodies. We've developed a series of those, and we've been able to use our humanized mouse models to evaluate those. So this is just some data. We've put a bunch of different human tumour antigens, looked at a bunch of different HLA-restricted epitopes, and just really to show that we've got lots of tools and toys that we're using and T cell lines and things to measure human antigen-specific T cell responses. And so this is with our in vitro cultured DCs, and we were able to show that CLEC9A targeting was superior to either DEC205, which targets all DCs, or non-targeted DCs in terms of inducing anti-tumour or reactivating CD8 T cell responses. We were also able to show this in melanoma patient PBMCs that we knew had a pre-existing response to the NY-ESO antigen. And I really think this data really kind of astonishes me still. I think it's quite remarkable because it really shows you the power of these DC1s in that there's so few of them there in the PBMCs, and you can see by the CLEC9A binding how many cells are actually binding to that compared to DEC205, which is everything's binding to. But even that small tiny amount from frozen PBMCs that have been stored for nearly a decade was still enough to really potently reactivate those CD8 T cells. So I think when we think about how many cells we need to add or how many DC1s, I think the answer is probably not very many. We wanted to go one step further in our humanised mice and look for antigen-specific T cell responses. So you don't really have this a really good naive human T cell repertoire. And in essence, what we were trying to do was make a human AT1. And so to do this, we just put a lentivirus encoding an NY-ESO1 specific T-cell receptor into the CD34s before we made the mouse. We get normal engraftment of everything else. And then within the CD8 T-cell compartment, we get a small, it's usually around 1 to 10% repertoire of naive CD8, so they're naive from CCR7, CD45, that express the NY-ESO TCR. So we think this is a really good model now that we can look at vaccination and amplifying of antigen-specific T-cell responses. Those T-cells are functional. This is just evidence to take them out, stimulate them with peptide, and they expand. They secrete cytokines, including interferon gamma, when you give them cognate peptide. And they also kill tumours that express the right HLA and the antigen. And so we'd use these just to look at whether we can actually prime using the CLEC9A and DEC205 antibodies in vivo. And so for these, we've been injecting the targeting antibodies in conjunction with poly-IC to activate them, also in conjunction with a FLT3 ligand to increase the DC numbers in vivo in these mice that are bearing naive antigen-specific CD8 T cells. And we've been able to get really nice priming in vivo and show that those cells have effector function both ex vivo as well. So now where we are is really trying to build those two models together to have both these naive antigen-specific T cells and the tumour in there. We've got a model that works. We're just really now starting to test it with some of these vaccine strategies. So that's just a summary of the CLEC9A. That data really helped move that forward into a data package for new IP, and that's now being developed for clinical trial. I was just going to flag that one as one other very small model that we recently published in collaboration with Simon Phipps that really shows the power of some of our humanised mouse models that we've been doing in other work, not just cancer. And this was a beautiful study that Simon just published in Immunity, where he showed that part of the propionate within the microbiome transferred from mother's milk was upregulating FLT3 ligand in the intestine. And that was increasing our plasmacytoid DC numbers, which was then giving enhanced protection against respiratory virus infection. And we didn't get as far as the infection in this, but all the rest of this, the microbiome and the propionate, we were able to replicate in our humanised mouse models and really show that some of these concepts are actually transferred over. I've whizzed through that. I'm sorry, I'm running out of time, but that leaves me just to thank everybody involved in our funding and collaborators. Thanks.
Thanks so much, Kristen. We've got time for a couple of questions, so let's fire away. Any from online?
Not yet.
No? Okay, I'll start. So I would like to talk about the mismatch human CD34 progenitors and the human melanoma, and you said it didn't really make a difference. So why? Is it really immunosuppression that's mediating that? And what if you tickle that model like with little activation stimuli or something like that? Would then suddenly you get faster tumor clearance?
So some of these models, we've only tried a couple of cell lines. You can do this with PDXs, and there's a lot that have been, there's quite a few groups active in this space. One of the things I think with the humanised mice, which could be an advantage or could be a disadvantage, is that they're inherently, really naive. So, and so I think the T cell, whether they're just, really at that immature stage, the same as cord blood, for example, it doesn't, you don't get as easy reactivation in T cells as what you do from adults. So you could argue there that that's just a really naive situation. But we don't really know. We know that, they don't express PD-1 or anything like that necessarily either, and I think that's why the checkpoint inhibitors don't work they work in some models, but not a lot. I think it's just a quite immature, naive system, which could have some applications, but not. If you're asking what we're asking, which is can you really boost that? What can you really kick to get that started? I think it's a good model. So, yeah.
Great.
Not sure if I answered your question.
No, yeah, I think so. Marie-Liese, hang on. wait.
Thank you. Yeah, I was just wondering about you had mentioned that these DC1s there were some that had a role in the tumor and in the lymph node to activate the T cells. In terms of the vaccine, do you think those DCs would have to go, where would they play a biggest role? Is that they need to go back to the tumor? Would you want those vaccine DCs to go to the tumor site or the lymph node?
A good question. I don't think we really know. And I think, so some of the stuff that's been done in the mouse where they've compared DC1s with monocyte-derived DCs, and they've also compared them intratumorally and IV. If you're going to load your DCs with antigen, I think they could probably go everywhere, but they work. The data there shows that they work in both locations, and so IV would be just as good, which would indicate lymph node priming. If you're not going to load, so the other ones have done intratumoral. So if you don't put antigen in there, then you really need to direct them straight to the tumour. So intratumoral injection works in that case, and presumably that's because they're picking it up and going to the lymph node, and there is a little bit of evidence for that. The other way around is that the MODCs on their own are known now that they don't directly prime the lymph nodes themselves. In the lymph nodes, they require antigen transfer to DC1s in vivo. So that's been shown in mouse models. Whereas the human DC1s can directly prime themselves. So in that case, that you're making the assumption that they can and will and should probably go directly to the lymph node themselves. But I don't think there's a good answer from that. And I think the more we know about the tumour site, the more important that's becoming. And of course, you either need to deliver them there or they need to be there in the first place, which they aren't in most cancers. So, yeah.
I think in the interest of time, we might have to wrap it up there, but let's just give a round of applause for Kristen for an impressive body of work. Thank you. No, it's good.